Okay, so PAICS is an enzyme that every cell has. Now, all cells need purines to support DNA synthesis, RNA synthesis and the like. It's a key part of the nucleotides. And most normal cells use the purines by resalvaging them when they break down the cells and they reuse it. But they can all make the stuff, as they say, de novo from the very beginning. And PAICS is one of the enzymes involved in that pathway. Now, most cells, as I say, just use the salvage pathway, but cancer cells, because they're growing so rapidly, need to make use of other sources of the purine, so they need to rely on that synthesis pathway. So the argument is, if we can block that PAICS enzyme, that enzyme that's part of that synthesis, you can slow down the rate of the growth of those cancer cells without hopefully affecting normal cells. When we started, we thought we were going to have quite an easy program. The enzyme itself is very well characterized, it's very well behaved, you can use it in biochemistry, you can run screens and everything else, and it has a very nice structure and pockets that we think you should be able to use to target. So we just set this up and ran it as a sort of normal high throughput screen, thinking we'll find lots of interesting things and be able to develop those on. And we found nothing. We ran that screen, we screened I think about 200,000 compounds and found nothing useful to progress further forward. But we still believed in it as a target, so we went to another idea, which is to use fragments. These are much smaller molecules. They're very good at finding pockets and holes in enzymes, so you can usually find things that will bind, but because they're so small, they're also very weak. So it means you have to then do a lot of chemistry to grow them, and that's what my group do. We're medicinal chemists. But in order to do that, we really need some form of information around the structure, how the fragment is binding, and that's normally a crystal, a protein crystal, and we were never able to get that in PAICS. So we had small fragments, they were binding, but the challenge was how could we take them further forward. So when we were in that position, we had some really nice fragments, but they were very, very weak. They were sort of 800, 900 micromolar, very, very weak. So what we had to do was build a computer model, an in silico model of the protein, base it on other structures that are known, and then we used a lot of biochemistry, a lot of structural biology. We did a lot of site-directed mutagenesis to change amino acids in those proteins, test them, see what difference it made, and use that to keep building that computer model, such that in the end we had a model that it wasn't a crystal structure, but it was almost as good as. And so we were then able to use that to design the fragment work, build out the molecules, and from those really weak fragments we ended up with something that was almost 100,000 times more potent, really potent small nanomolar molecules that, as we speak now, we're looking at in vivo models to see whether it really does what we think it should do.